Quick review. That we did our little candle experiment where I lit a candle in the middle of the room, and we noticed that the smell, because it is basically molecules dissolved in a solution of air and a bunch of stuff, that smell radiated outward from where it was, which follows those laws of diffusion that say molecules dissolved in solution will move from high concentration to low concentration until there is no high and low concentration, which is what we call equilibrium. Things are equally distributed. And that it turns out some small molecules like oxygen and carbon dioxide and water can do that right across the cell membrane. So those are kind of the easy ones. And I mentioned Beaker problems, I think. Here's what Beaker problems look like. And you're going to get a page of them today. Basically, what I do is give you a beaker on a piece of paper with water in it, and usually a membrane. And you can sort of consider that to be like a giant cell membrane. And then I give you a solute, and I show you where it is, and I tell you what that solute is and whether it can cross the membrane or not. I'm only going to use 4 solutes in Beaker problems. Oxygen is one of the ones that is small enough to cross the cell membrane. So if you see a Beaker problem with oxygen, you can basically just pretend the cell membrane isn't there. And the question in a Beaker problem is always what goes where. So in this particular Beaker problem, somebody want to tell me what would go where in the very next instant? What kind of motion are we going to see? What's going to spread out? The oxygen, which is in this beaker, all concentrated outside the cell. And it's supposed to go from high to low concentration. Which way would that be? Armani. That's it. You got it. From outside the cell to inside the cell. That's high to low, right? This is high, this is low. And so what's going to happen is the oxygen is going to move into the cell until there's no higher or lower concentration, until it's all equal, right? But we don't care about later. We want to know just in the very next instant. And what's going to happen in the very next instant is the oxygen is going to go into the cell. Brady, how about the second peak? So high to low in this case, as far as what goes where, means high is here, low's out here, and so the oxygen is going to leave the cell. Does that make sense to everybody? Okay, draw those.
You want to draw the text too?
Yeah. Those are the easy ones. Those are the Beaker problems. If you know it's something that can cross the membrane, it does. It goes from high to low concentration. You can basically pretend the membrane's not there. When it gets tricky, and this is going to be the bulk of what we deal with for the next couple of days, making sure you understand this, because it's not intuitive. When it gets tricky is when there is stuff that is stuck, that is too big to cross the membrane. And when that happens, you'd think it just stays put. But what happens instead is that stuff being stuck, solutes being stuck on one side of the membrane or the other, inside or outside the cell, sets up a kind of pressure that makes water move. And that process of water diffusing across a selectively permeable, that just means some things can cross but others can't, membrane is called osmosis. A much misunderstood and misused term, osmosis. You put your head down on your textbook. Someone says, what are you trying to learn by osmosis? That's a misuse of the word osmosis. It would be diffusion if you were somehow soaking up words from a book. Osmosis is always water. And to figure out which way the water is going to move is the challenge, because it's weird. It's not exactly what you think. What happens is, if those molecules are stuck, you immediately have to switch the way you're thinking about and those molecules are stuck, what's going to happen is the water is going to move. And again, there's two ways to think about it. It's going to move from high to low water concentration. But it could be hard to think in terms of water concentration. So the other way to think about it is it's going to move from low to high solute concentration. In other words, it's going to move the opposite direction that the stuff would have wanted to move. Anybody need more time? Rather than try and explain this without something to look at, let's just do a beaker problem, and I'll show you what I mean. Remember, high to low water concentration, low to high solute concentration, which turns out to be the same thing. Now, don't draw these yet, but new beaker problem is now it's not oxygen anymore. Now I'm telling you it's sugar. Sugar is one of the 4 solutes that I'll use. I'm telling you sugar's too big to cross. It's stuck. So if there's a bunch of sugar stuck outside the membrane, we now have to stop thinking about which way the sugar moves because it can't. It's stuck. And what that does is set up water pressure. It sets up osmotic pressure. We got to flip our brains to thinking, okay, now what we said is water is going to move from high to low water concentration. Where in this beaker is the water more concentrated? That's pure water, right? That means that's the most highly concentrated water. So if water is going to move from high to low water concentration, which way is that? From inside to out, yeah? Okay. Now, the other way to think about it is that the water is going to move from low to high solute concentration. In other words, low solute, no solute really, to high solute, or basically the opposite way that the sugar wanted to go. It's almost like, another good way to think about it that goes along with that second thing is it's almost like the water is compensating. It's trying to dilute the sugar. Okay, so yes, what goes where? The water goes out, and in fact, when there's water movement like that, we name the solution. And the name of a solution that water leaves the cell in is hypertonic. It is a hypertonic solution. Okay? So if that's the case, if it's when the stuff's stuck, the water moves from high to low water concentration or low to high solute concentration. Second beaker. What goes where, Abby 6? Second beaker, what goes where?
The sugar molecules go in.
Sugar can't go anywhere.
Sorry, the oxygen.
There's no oxygen.
The water molecules go on. Sorry.
Ah,
The water molecules go.
there you go. Water is going to go from where there's more of it to where there's less of it, right? Water goes from high concentration outside to low concentration inside. And by the way, the word we use for that kind of solution is hypo. That's not a dumb question. That's my favorite question. That's the question you should ask. Mr. Sandro, if sugar can't cross the membrane, how did it get in there in the first place? Yeah. So that's next. After we get the hang of osmosis, we'll talk about how cells do get things that are too big to cross by diffusion into or out of it. I'll give you a hint. A lot of it has to do with those proteins we talked about early on, the transport proteins. If you're like me, and you are, in that those two words, hyper and hypotonic, seem like they're going to be hard to tell apart. I always had trouble with that when I learned this stuff when I was a kid. So I'm all about dumb ways to remember stuff. So my 12-year-old brain is what I usually call on for stuff like that. And my 12-year-old brain looks at those two words and sees that the difference between the two words is right there. And my 12-year-old brain goes, pee. It's like the cell has to pee. Water comes out of it. That's literally how I've remembered it for 25 years, is that hypertonic solutions make the cell pee. They make water come out of the cell. If that works for you, use it. If it doesn't, find something that does, because these two are hard to tell apart. Okay? Questions? It's hard. Okay, it's simple at this level, but you still don't believe it. Like the reason that I just had you take an egg and put it in a cup and cover it with vinegar. And remote kids, I told you to do that yesterday. If you didn't, you should do it now. You took an egg and covered it with white vinegar. Why? What's that going to do to the egg? Riley.
Doesn't it disintegrate the shell and leave? Because there's a second shell on the egg and it just leaves like the second. Like I don't know what the exact word for it, but it leaves like the second shell.
So the shell, what we call the shell, the hard part,
Yeah.
is hard because of calcium.
Yeah.
And vinegar is a weak acid and knocks the calcium right out of the eggshell. It's going to bubble up, and you're going to see that shell come off and leave what we call the egg's membrane. Membrane. You've just made, basically by tomorrow, a giant Beaker problem cell. And we're going to do stuff to it to prove that osmosis works. Your first page of Beaker problems is here. It is due tomorrow. The answer is always what goes where. You don't have to name solution types yet. If you didn't just get one, grab it on the way out. It's on top of the eggs right here. Say it again. Yes.
With the following
You got
students,
14?
please come to the main office. Sydney Small, David Thomas,